Before we started, I would say, so the format of this presentation is that we probably can take questions right in the middle while we are talking, because we are pretty familiar with the topic and it's easy for us to answer, but we know a little bit less about you. So it would help us to really navigate of how we describe a particular problem if we get the questions as we go. I can say just a couple words about myself to start this presentation. So basically, my background is in drug discovery and biophysics, or at least it started that way. So I did my undergrad back in Ukraine, and afterwards I moved on something called Fulbright Scholarship. And I stayed for a year and a half with Dr. Raymond Skenazy, which in an antiviral discovery lab. So that lab was most famous for discovering hepatitis C cure, which is sofosbuvir. And for a year and a half, I was working trying to discover drugs for hepatitis B using biophysics and simulations. And we did discover like some interesting compounds. And next year, I spent with Professor Gil Alterowitz at Harvard Medical School. And then I almost finished my master's at MIT. And then I went to do an internship with Professor Yoshua Bengio. And now I am in the process of transitioning to my PhD. And my main work here is I'm organizing molecule discovery groups, and I'm trying to coordinate different projects with each other.
Thanks a lot, Maxime. So hi everyone, it's a pleasure to be here, and thanks to the organizers for hosting us. So I'm Paul Bertin. Very briefly, I'm a PhD student at Mila, working with Yoshua Bengio. So I'm from France, have a background in mathematics and machine learning, and I moved to Quebec 2 years ago. And I started working on medical applications of machine learning. So I worked a bit on computer vision. And right now I focus on gene expression data and drug repurposing for this project. And my research plans are to try to adapt like causal inference techniques to gene expression data. And yeah, it's a pleasure to be here. And I will let Maxime present the first part of the talk.
Yeah, sounds good. Next slide, please. So I wanted to give an overview of basically an overarching objective of Mila Molecule Discovery Group. So it's a pretty big group. I think it consists of right now about 10 advisors, and there are more than 30 students involved part-time, and I think about 10 of us full-time, but we are expanding. And basically, what is our general direction, right? So what are we trying to solve together? And it's just one single topic, which is how to better search in the space of molecules. And under search, I mean both discovering new molecules, right? So which is basically generating molecules atom by atom or block by block or in reaction space, whatever that would be. But it also finding a new drug combinations, right? Which is basically what are the right dosages, right? How much of each drug we want to achieve an optimal effect on cells and in like later proxies of human body, right? And yeah, so this two combine together, and in the future, these two directions will actually fully merge, because I believe the future of drug discovery would be to be able to discover one drug that probably targets more than one target at the same time. So we definitely are very much from the very beginning interested in that direction. So just to give a very broad overview of the topics that we are working on, right? There is one family of topics that is really, really interesting to us, and this is how to make reinforcement learning to search in the space, right? So how to develop reinforcement learning for search. There are not too many published papers. So if you look at molecule de novo design, right, then we have things like MolDQN or molecular PPO from Stanford group, right? So, and also we have earlier presentation today from groups like different startups like NVOAI, and there is Atomwise and others. But these are more of applied reinforcement learning algorithms applied to the search, right? We are, as academia, we are trying to come up with a more fundamental way of just writing algorithms in such a way that from the very beginning, these algorithms are designed to do search. And what does it mean, right? So let's say having a fixed compute budget, right? Let's say having a fixed compute budget of, let's say we have 50 GPU days for the project for one protein, right? And we want to design the best drug, right? Usually the problem in RL formulation that we want to have an algorithm, right, which have something like a Q value function or Q function, which says, okay, given this settings, right, given the state, predict the optimal action to take given the value of the next states, right? But what we are really trying to do here is we want to go a little bit beyond that and abstract from this usual reinforcement learning paradigm, right? And we want from the very beginning try to write a function or a formula which would say, okay, so what is the formula we have to optimize for to achieve the best possible objective, which is in search, given a limited computational budget, right? And this requires a lot of thinking. And this is a very long-term objective, which is not so easy to solve, and it will probably take more than one paper to come up with a solution. Moreover, it's probably not a single solution to this. The next very big set of challenges, or the huge challenge that we are trying to solve, is that it's very, very recurrent, no matter where it is. Is it drug combinations, or is it generative molecule discovery? There is a very non-classical phenotypical machine learning scenario where we have different oracles, and these oracles are so different in their cost of running. For example, if I am in a reinforcement learning setting, I can do a very cheap estimate of molecule energy. So I can come, and let's say some reinforcement learning agent comes with some molecule proposal, and it proposes some molecule graph, and we can just run message parser neural network and evaluate this graph and say, okay, here is the energy, so whatever the distribution would be given uncertainty, right? So this is very, very quick to evaluate. But if you look, what's the next level of oracle? The next level of oracle would be something like docking. So docking takes seconds. And let's say, okay, what's the next level of oracle? And the next level of oracle is DFT. And DFT is dimensional function and theory computation to parameterize the molecule, but also something like free energy perturbation, right? And this kind of simulation is already 1 day, right? So looking at these 3 simulations, right, it's already 3 orders of magnitude. And if you go over, right, if you want to synthesize a molecule and test it in, let's say, in biological assay or in cell, so it's usually the speed of this kind of thing is limited by the production of how many molecules they can produce. And then chemists can only produce, let's say, 3 molecules per week, right? So they usually can take 3 molecules in parallel, and they can produce 3 molecules a week. So now there is basically 3 orders of magnitude, 3 orders of magnitude, and 3 orders of magnitude bigger cost. So usually machine learning methods or active learning methods as we see them, they usually are set up for difference in costs, but not really 3 orders of magnitude difference. So we really have to come up fundamentally with the new ways that allow us to incorporate feedback, right? As we are generating molecules or molecule combinations, right? We want to come up with a way that will tell us how to run these oracles of so different costs. And right now, fundamentally, this problem is not solved yet. And the next thing that another big set of challenges that we are trying to solve is basically what are the mathematically solid foundations, right, to simulate biophysical systems in general, right? To give you an idea, right now there are a lot of papers on, let's say, graph representations of molecules. And there are a lot of papers, okay, we can represent molecule as graph, as string, as point cloud, as wave, but there are so many of these representations. So, and what we are really trying to do here is think, okay, what is a very fundamental way to represent molecules, right? Because we want to get molecule energies, right? That energy could be energy of a particular conformation. That energy could be energy of a particular conformation in the context of protein binding. That energy can be just basically integral of many conformations that we want to compute at once, which will basically describe something like free energy, right? And we want to come up with a certain way to simulate molecule, but there are so many of these representations, and there are so many papers which each of them claims to have better results somewhere. And fundamentally, is molecule a graph? Is it a point cloud? Is it something else? And what we are really doing here, right, we are developing a fundamentally very new kind of representations, right, which are kind of neither graph, neither point cloud, neither something else, because what we are doing is we are going for the very first physical principles. And looking at this, we can say, okay, what are the most important properties that we really want to have for a molecule? And these properties would be something like okay, when we rotate a molecule, the energy doesn't change, right? So we do want to have rotational equivariance, right? And in terms of energy, this will be rotational invariance, but in terms of force, we really want to have rotational equivariance, right? Which is basically when we are rotating a molecule, the force vector should rotate the same, or something like a dipole moment, right? Vector properties should rotate, right? So we want something like this. What we want next, right, is we also want to be Lipschitz continuous. And what does it mean to be Lipschitz continuous? So whenever we perturb coordinates of the molecule a little bit, right, a tiny, tiny bit by infinitesimal amount, we want the energy to also change a little bit, right? And this is very important because if we want to have 3D aware simulations of molecules, we really have to have something like this, right? And there are also other key challenges, right? So how to propagate information within a molecule graph or within the molecule point cloud or between atoms, right? It's fundamentally a challenging problem because if you look at usually the data sets that are more used right now by machine learning community, this is data sets such as QM9. Luckily, there are efforts by pharma or by startup or pharma companies where they try to be a little bit more biologically inspired. So they do evaluate on zinc and there's some more drug-like molecules. But the problem there is we really want to be able to pass a signal or information, right? Thinking about how forces work, right? We have some long-range forces, which are electrostatics and things like this. But we have also short-range forces, which are bonds and bond rotations and angles. And we need to incorporate all of this. So our, I would say, very key paper, which is, I would say, the very first step of us in this direction is something called TPNN, right, which is a tensor passing neural network. And what this tensor passing neural network specifically does is that instead of passing messages which are vectors, right, it passes messages which are 3D aware, which are tensors, which describe deformation, second order deformation, right, and they equivariant with rotations, right. And then we also basically, in terms of the graph solving problem, in terms of the signal propagation problem, right, we have different versions of sending the signal. So we have certain amount of signal traveling from atoms which are far away from each other, but there is really a low bandwidth. And we also have certain hierarchy, right? So the molecule is represented as a hierarchy of blocks. So very much like was initially, I would say, in machine learning that was first introduced by Regina Barzilai, but it's an incredibly efficient method, right? So this is kind of representation we are working on. And now today we will talk about one specific application and more specific about specific implementation of one specific application. So to say what it is, right? What we really want to do, oh wait, back, I still had that slide. Yes. So this is a full cell simulator. So, and why do we need a full cell simulator, right? And there are many reasons for this, but in particular, what we're interested in, we have seen, let's say, cancer drug combinations, right? And we know that for women, right, just probability of developing a breast cancer is about 10% in the United States during the lifetime. So that's a lot. And we found this study which really, really inspired us of drug combinations predicted by a huge panel. So there was a study by NIH, but there were also a bunch of startups like Grail and others and Foundation Medicine participating in that. So basically what they did, they said, okay, we will put a huge panel of physicians, and this would be called a cancer discovery panel. And it's a huge panel, it's like 35 people. And basically what they will do is they will describe a drug combination for a stage 4 cancer drug patient, right? The other way we will treat them with a standard of care. And the study was called I Predict, and the results were released about half a year ago. And there is a new one, which is much bigger, which is currently running. And what they have found is that if they have this huge panel of physicians, if in the control group of stage 4 cancer patient, none of the patients survives, then when they have a panel of physicians under incredible amount of effort and looking at all this molecular diagnosis, they actually about 20% of patients are able to survive during the course of study, which is about 18 months. And it's statistically significant, and there are also other like good correlations, like for example, tumor keeps to be the same, right? So the size does not decrease. the size of the tumor is stagnant in many more people in the group which receives the combinations described by patients. So we were really, really interested in this. So we are thinking, okay, so what can we do in order to build? So because we cannot test a lot of combinations of drugs, right? What we can do to build a really good cell simulator, right? And some sort of problems which we encountered. First, if you look at like drug combination, like current drug combination models in literature, the models like Decagon, they're written in TensorFlow, unfortunately, really, really, really slow. So basically just running Decagon, which is published by Marian Kazitnik and Jur Leshkovitz group. Running that on the same data set, it takes about a week on the data set of just predicting drug side effects, right? And one of the our very first challenge to even get started was how to write this efficiently. So what we did is we moved the full data set on the virtual RAM on the GPU directly, and then we started doing computations there. So from the time of about a week to 10 days training, we transitioned to 4 hours time of training. Now we can run some experiments, right? Then we did basically, this was mostly published, but we just happened to apply already published algorithms to a problem which we really want to solve. So there was a method called large-scale LRGA, which is large-scale global attention approximation. And this appears to be very, very important to have a graph like this with a high degree of connectivity to be able to efficiently pass the information, to basically have certain feature vector, which will represent the global state of the graph, which will be used for conditioning on the message passing. Another set of challenges is that there is so much data now. So if you look at cells, so there is so much data. There is transcription binding factor like ChIP-seq, and then there is binding, drug protein binding. Then there is functional things like CRISPR interactions, right? So what we did for now, we at least managed to integrate in our simulation pipeline, we integrated transcriptomics pipeline, right? So our model can take transcriptomics as an input, and we show that it's very, very efficient. And finally, then basically we started looking as, current graph models for synergy predictions. And then we just figured out that, it's not even correct to represent models the way they are, because when we have a system, right, so what we want to simulate is a cell, and we are simulating a perturbation on the cell. And the way the graph eroding model work is that basically they have many, many, many different drugs, and all of these drugs want to pass messages to all of these proteins in the cell, right? And then simultaneously, this graph somehow wants to predict all of the pairwise drug interactions, right? So all of the drugs are present in the cell at the same time, and at the same time, we are trying to predict all of the possible synergies. So this is incredibly complicated to learn. So basically what we did and currently are doing is redesigning the framework of how graph learning happens for molecules. And that basically includes something we call signal on a graph framework, right? Or the simulation framework, where we basically have a simulator, right? Which is a graph which exists in a certain state. And then we are adding two drugs, and we are able to simulate it and predict the outcomes. But with the same system, we also able to model other perturbations, right? Such as, for example, gene knockout. And finally, the most exciting part for me is active learning on cells. So of course, there are many, many possible implementations of what we can apply drug combinations to. Right now, we concentrate on two. One is drug combination learning with Mike Tyers at IRIC, and this is our long-term collaborator. And basically, there we are using cells to predict new efficient cancer combinations. But also there is another one, which is basically drug combinations for COVID-19, which we really, really need now because it's a very urgent problem. And the rest of this talk, and while I'm speaking, there are already cell experiments running, so this is kind of exciting. So we are running our first early cell experiments as we talk. And the rest of the talk, we will focus on a very specific application, which is how we can use drug combinations to target COVID-19. Next slide, please. So this work is performed thanks to our collaborators. And we are funded by Bill and Melinda Gates Foundation, and we work very densely with relation therapeutics. And this work, of course, wouldn't be possible without them. But today, as I am representing Mila, I will represent the rest of this talk will be on the results that we did, specifically machine learning models that we did inside Mila. Next slide. Next as well. Right. So this basically is a very generic slide. So this is basically usually the process of drug discovery. We talked about de novo drug discovery, right? And thinking into the context of COVID, we have an urgent problem, right? Which is basically we want to be able to do something for COVID now because it's a very huge problem, and de novo drug discovery will likely take a very long time, right? So as I have seen, there are important drugs that are right now being used in clinics, and there are two success cases. One of them is remdesivir, right? And remdesivir is a transcriptase inhibitor developed for some other virus, and there have been extremely promising clinical trials data where they show that patients who take remdesivir recover faster. And another one is dexamethasone, which is a hormone. And I'm not exactly sure why it works efficiently, but as I understand, it reduces inflammation. So basically now we are thinking, okay, we found one drug and there are a bunch of other, like there was recently by another study founded by Bill and Melinda Gates, there was another drug which seems to be even more potent, so-called apilimod, which is a failed drug for rheumatoid arthritis, which seems to inhibit COVID-19 on cells by far better than remdesivir. So now is the question, okay, so apilimod is moving up the clinical trials, they did their mouse tests. So what can we do as a machine learning community? What can we do for COVID-19, right? And what we are focused on is basically we are trying to discover new COVID-19 drug combinations. We likely will include remdesivir, but also one of other 25,000 drugs, and maybe two of other 25,000 drugs already approved to be taken by humans. Next slide. So what we are looking for is so-called synergy, right? So in this particular example, we look for two examples, and synergy means is whenever we have two drugs, the two drugs together are more than each of the individual ones, right? So we have an added effect. And this is something that we would be looking for. And in this case, we assume there might exist a combination of two drugs which is more efficient than a single drug like remdesivir or apilimod alone. And what is shown on the slide here is basically if you have two concentration of a drug, usually when we add two drugs in the cell, right? And as now we talk in the context of a cell, all the rest of the talk is in the context of a cell, because there are also synergies on the level of the organism. But right now we'll just talk in the context of a cell because that's something we can do and validate very quickly. So basically, if you add two drugs, we expect the combination or the combined effect of them to be linear. And sometimes the two drugs can prevent each other from working. And I can give example from a cold, right? So to cure cold, there are two different ways to cure a cold. Either we increase the number of cough from a person, right? So this cough goes away, or we basically trying to drain the bronchs, right? So we try to take liquid in and then person doesn't cough anymore, right? And these two would be antagonistic, right? And here, what we are looking for is a synergistic drug combination, right? And there are a lot of examples of this in clinic. If you look at HIV or tuberculosis, these are multi-drug combinations that are applied to the treatment of patients. And that's something we are looking for COVID. Next slide. And so what we are doing here is if we try to take, we just basically, if you're looking at the time that we need to try to look at all the pairwise combinations, right? The COVID appeared very recently, right? And there are in so-called Reframe library, which is all FDA-approved drugs, which exist as a huge library, which is funded by Bill and Melinda Gates, which is one of their major funding effort for which they invested probably about 50 million to just make all of those drugs and store them together. To search just a pairwise combination, not a triple combination, pairwise combination. If you screen for 5,000 molecules a week, which is actually something we cannot do, this is the most optimistic estimate one can get, because this COVID-19 cells, they require BSL-3 lab, and this is extremely expensive and extremely low throughput lab. then it will take 300 years to search for all the drug combinations. And this is not something we can do. So this is way too slow. We need something that we can do in probably months from now. So what we do is we apply machine learning to basically try to search this space. And we do it in an active learning setting. Next slide. And basically here we have this combination of, right, one, we want the agent to learn as much as possible about the space of possible molecules, right, which is basically tries to say, okay, which combinations it didn't try before, which combinations by trying more of them it will learn most. And this is a setting which is very well explored in Bayesian learning. And another thing, it also wants to be greedy, right? Because it has a limited number of trials. With a limited number of trials, we want to achieve the best possible effect, right? And basically training this agent, which both explores and learns about the space of possible molecules, is the problem that we are trying to solve. And Paul can take from here and show a little bit more practical results that we actually have been able to achieve.
Yes, thanks a lot, Maxime, for the presentation. And do not hesitate to ask questions at any time. We'd like to have it interactive. I'm not sure if you can unmute yourself, but feel free to ask questions in the Q&A, and we'll make sure to answer them during the talk. So as Maxime explained, it's a big challenge to explore the space of all drug combinations. And the question of how to efficiently explore the space of drug combination is complicated. And here we'll present like our approach to solve this problem, which involves machine learning and active learning. So basically, this is like the big overview of our proposed pipeline. And as you see, it's like the lab where like compounds are tested and a machine learning pipeline, which interact together in a loop. So basically, experiments will be performed in the lab on some drug combinations. And then the results will be sent to the machine learning pipeline, which will be in charge of recommending the next drug combinations that will be tested in the lab. And this is expected to take like around one week per group. And we expect to have the experiments running in a few weeks from now, and it will last for a few months. So we'll be actually able to test lots of combinations. And today, we'll specifically focus on the machine learning part of the pipeline, as this is what Maxime and I and others have been working on. So this is a zoom in the machine learning pipeline, which has like three main components. So first, the data, which is like a very important part, of course. So the known synergies for some of the drug combinations. And this part will grow along the time when more experiments are performed. And we also have other kinds of data which correspond to more general biological knowledge, such as fingerprints, genome ontology, drug targets, protein-protein interaction, and differential gene expressions. We'll go over this in more details later. And then we'll aggregate all those data modalities, and that's a big challenge to be able to aggregate all this data together and use it in one machine learning pipeline. And our proposed solution involves graph neural networks, as some of those data have a graph structure. And the graph neural network will be in charge of predicting the synergy or the antagony of drug pairs, unseen drug pairs that haven't been tested so far. And this model also needs to model uncertainty, as this is a key component to be able to explore the space of drug combinations efficiently. And the third component of the machine learning pipeline is what we call acquisition function. And that's the part which is in charge of deciding which combinations will be queried next and suggested to the lab so that they perform experiments on those specific combinations. And now I will describe in more details the data that we use. So some of this data is like used just as the purpose of training our algorithm before the data is acquired, such as the differential gene expression, for example. But other data will be used also during the main, while the main experiments are performed. So there are four big parts. The first one is the protein-protein interactions, which model like the interactions that happen within the cell between proteins. So those interactions can be of many types. It can be protein-protein binding, for example. It can also be scrapped with NLP from the bioinformatics literature or the biology literature. We use four different databases which are publicly available, namely String, Snap, Hurry, and BioGrid. And another key component is having information about the drug. So for now, it's quite simple. We use Morgan fingerprint, which is like a widely used tool in bioinformatics. And that's one of the components on which we could improve on the future in the future by using fancier methods that are being developed right now in other parts of the project Maxime mentioned earlier. And we also have access to the drug targets, that is to say, basically the proteins with which a drug is expected to bind. And that's also like a key information to have in order to be able to predict the synergies between drugs and what's the effect of a given drug in the cell. And for that part, we use two different databases, the Drug Repurposing Hub and ChemDL. And the last component is differential gene expression, which we expect to acquire at the same time as the drug synergies when the experiments are being performed in the lab. But for now, we use the CMAP L1000 dataset as a way to train our machine learning pipeline beforehand. And basically, what is differential gene expression? More specifically, it measures the concentration of mRNA corresponding to different genes in the cell. And the higher level, the higher the expression of a gene is, the more this gene is used in the cell. That's a very brief explanation, but that's the main idea. And basically, we can then compare the difference in gene expression before and after a given drug has been applied in a cell. And that gives us a very nice insight into what's the effect of the drug in a cell. And we hope to leverage this insight in order to better understand the synergies of the drugs. Now I will describe the graph neural network pipeline that we came up with. It's a bit technical, so maybe some of you are not very familiar with the like details of machine learning, but like please do not hesitate to ask questions, and I'll try to have to present the main ideas of the pipeline. So as I mentioned, some of the data we use, namely the protein-protein interaction networks, as well as the drug targets, have a graph structure. So we use GCN, and the graph that we consider is composed of both protein drugs and protein nodes, sorry, and drug nodes. And so our graph has two types of nodes, so drugs and proteins. And the fact that there are two types of nodes, which have really correspond to different things, made us use what we call multi-message GCN. That is to say, compared to a regular GCN, which send messages between nodes, here we have different types of messages for different types of connections. So we have messages for corresponding to drug target interactions, and we also have messages for protein-protein interactions. So we also have self loops, which are in charge of propagating the embeddings of the different nodes across the different layers of the network. And in order to better understand the long-range interactions within the cell across the network, we use a low-rank attention, which is like a slight variation on the regular attention mechanism that some of you might be familiar with. And it allows to scale the attention mechanisms to many more entities. Here, if we use, so for now we use attention between drugs, so a few thousand nodes, but we are planning as well to use attention between proteins, and it's around like 20,000 nodes here. So it becomes very tricky. And compared to the basic attention mechanism, here there is no sigmoid softmax function, sorry. So basically it allows us to compute the matrix products in a different order. And it means that the big attention matrix is never explicitly computed, and that makes it much more tractable. And finally, in order to predict the synergy of a given pair of drugs, we use a predictor, which is like a basic MLP that we can condition based on the cell line on which the experiments have been performed. So we have different ways to condition the MLP. It can be film conditioning, which is like a specific technique to condition with a few parameters each layers of the MLP, or something like more basic, like shared layers for all cell lines, and then cell line specific layers. And another key component of the pipeline that I will describe briefly here is to estimate uncertainty. And it's a key component of the pipeline that allows us to address the exploration-exploitation dilemma that Maxime described earlier. And that's typically achieved in what is called a Bayesian learning by having a prior distribution on the weights of our model. And what it allows us to do is to predict the whole probability distribution of the synergy for a given pair of drugs, instead of just having a point estimate of the synergy, like just one synergy predicted, for example, but to have a whole distribution. And that's a much richer information that we predict here. Without maybe going too much in the details, there are lots of problems that arise when combining Bayesian learning to deep learning. Depending on the different techniques, it can be like untractable if we try to do something very complicated, or some people have tried to make simplifying assumptions by having like simple posterior distributions on the weights, but it wasn't working very well. And one nice approximation that has proven to be very effective is Monte Carlo dropout, and this is what we use in our pipeline. There are other ways that we could deal with this problem. We could use ensembles, and that's also a direction that we are trying to explore. And very intuitively, for uncertainty, what is uncertainty? There are many ways to define it. One way to define it is to consider that it's the variance of the probability distribution of the synergy of a given pair of drugs as predicted by the network. So if we haven't tested a given drug yet, we don't really know what will be the synergy, but maybe we have some idea. And if we don't know at all what the synergy will be, the variance of this probability distribution will be high. That's for the main intuition. All right, and now I will talk about the third component of our pipeline, acquisition functions, which are a key component to direct the search of the drug combination space. So very briefly, what is an acquisition function? It's a function that computes a score for each drug pair given the probability distribution of the synergy given the drug pair, which is predicted by our graph neural network described before. There are different strategies possible. So for example, just the greedy acquisition function is very simple, and it's pure exploitation and no exploration at all. That is to say, you just the score is just what's the expected score synergy for your drug pair as predicted by the model, and then you will query the pairs that have the highest expected synergy. You have the opposite approach, which is random pure exploration. In this case, the acquisition function is just a random generator, and it means you will select at random the next experiments to perform. So that's of course not optimal, and those are the two extremes. And then there are like fancier approaches which are in between the two extremes, and that try to take advantage of the uncertainty in order to explore a little bit while driving the exploration in the right direction. So there is expected improvement in which basically you want to focus on the drugs for which you expect to improve the most compared to the best drug combination that you have seen so far. And as described before, it can be estimated quite simply using MC dropout. There is another approach, which is GP-UCB. And here, so it's a bit different from expected improvement, as we do not focus on the improvements, but we will focus on just the drugs that have a high uncertainty, that is to say the standard deviation of the synergy distribution. And you see that compared to the greedy strategy, you have an additional term which gives more importance to the pairs for which you have a high standard deviation, that's to say like a high uncertainty. So I will present briefly the results, but of course that's ongoing work. So this is first slide about early experiments that we performed on cancer data from the DrugComb portal and the L1000 dataset, which are both publicly available dataset and which do not correspond to COVID data, but it's also like experiments performed on cell lines, and it has many similarity with the problem that we want to address in the end. So here we just wanted to explore the importance of the different features that we use. That's quite insightful. As I described, there are many different data modalities that we use. And for those specific experiments, we just use a simple MLP. There is no machine learning, no active learning, here. And we try to predict the CSS score, which is a sensitivity score. And we also, so this is not shown here, but we have many different scores that we can predict, synergy scores, such as HSA, BLIS, ZIP, for those who know a little bit about those things. And you can see that overall here, we have a pretty good fit with 0.8 R squared. That means that we can explain 80% of the variance that we've seen in the dataset with our model. And you can see that lots of features like the protein, the drug targets and the fingerprints explain well what we see in the data. And we can also see that when we use differential gene expression, the model performs even better. And that means that the insight provided by the gene expression data is quite helpful for our pipeline. Then we want to evaluate the efficiency of the exploration strategy using different acquisition functions. So those are more recent results, still on cancer data. And we use the multi-message GCN that I described earlier, with like the predictor is an MLP with a bunch of shared layers and then cell line specific layers. And what we see on that plot is the proportion of the top 1% synergistic combinations in our data set, which have been discovered by the pipeline so far along the steps of active learning. So you can think about the x-axis as time, and the y-axis is the proportion of the best synergistic combinations in our data set that have been discovered. So you can see that the very naive approach, which is random acquisition, which means that you just perform experiments at random, and it performs quite poorly, of course, compared to the other one. And then when we explore the space greedily or using the fancier acquisition functions, we discover much quicker the best synergistic combinations, and it's in like a few steps, 8 to 10 iterations, we discover almost all of them. One thing that we see here is that the expected improvement and GPUCB do not perform much better than the greedy acquisition. That may be like one bias that there is in our experiments, because in fact our data set is rather limited in size compared to all possible combinations. And it means that in fact, if you perform greedily just among the examples that are in our data set, you will perform quite well and you won't get lost in the same way that you could get lost if you had all possibilities available. So maybe in the real case, so of course that's early experiments and we still need to improve on this, but in the real case, we could expect this bias to disappear somehow and the greedy acquisition to not compare as well to the other approaches. So in conclusion, that's I think this project is a very nice example of how machine learning and the experiments being performed in the lab can interact together in a nice loop, as we saw earlier, and that's very exciting to me. So that's ongoing work. There's still some efforts to be put on improving the model and performing more tests on our current pipeline. In terms of research directions in the middle term, long term, there are lots of very exciting directions, such as improving on the graph representation learning part, as Maxime mentioned a little bit, and as well on out of distribution generalization. As you see here, because we use active learning, the distribution that is seen by the model is not static, and the model will explore new regions of the space along training. And also the fact that our model generalizes well far from the data that it has seen so far is especially helpful here, because we use those predictions to actually go and see what happens in that region of the space. So thanks a lot to the organizer for hosting us again, and I will briefly present the team. So that's a project in collaboration with between Mila and Relation to Repurposing, and it's funded by the Bill and Melinda Gates Foundation. So the leadership of this project, so there is the Professor Yoshua Bengio, Dr. Charles Roberts, and Dr. David Roblin. And the Mila team, which focuses on machine learning. So there is Professor Yoshua Bengio, Maxime Korbliov, Jerry Director Brooks, Isabelle Lacroix, and myself. And on the recover side, there is Charlie Roberts, Jake Taylor-Kings, Benjamin Swerner, Christian Rejep, Thomas Godley, and Jeremy Haters. And that's a pleasure to work in this project. And again, that's very exciting. Thanks a lot. And please feel free to ask any question.
Yes, I have a question from Yoshua, which I can read. You equated uniformly random search with pure exploration. I don't think this is an appropriate match. In my mind, pure exploration would just be knowledge acquisition like child play, not caring about exploitation. Oh, I see.
So we can.
Which is good at some very particular sense.
To say pure exploration is the same as.
So pure exploration would be just knowledge acquisition, like child play, not caring about exploitation. Okay, sure. In the sense of achieving some extrinsic reward, like discovering a molecule, which is good in some very particular sense. Yeah, but yeah, this is possible to do this baseline. So basically, to paraphrase this baseline, it would be if we have something like purely like bold, so which is batch active learning, to improve and sort just have it pure in exploration. Yeah, so we actually didn't have that baseline. It would be good to have, because right now we only had random exploration, but not something like pure exploration. So we had random acquisition function, but not pure exploration. That's indeed a pretty good point.
So I guess here, with the, regarding the analogy with a child playing, here there is no, it's an interesting comparison. Here there is no like really like time dependency. I mean, there's time dependency. Yeah, I'm not sure to precisely understand what you mean, Joshua. But yeah, random. We could describe it as the.
Yeah, exactly. So Yeshua says,
Okay.
child look in the places she doesn't know. Yes, exactly. So this is basically like bold. Yeah, tries to understand causal effects. Oh, but causal would be way different.
Actually, that's one very interesting directions that we could follow. That is to say, instead of having an acquisition function which is driven by uncertainty, it could be like an acquisition function which is designed in order to query the examples which are expected to give the most useful information from the causal point of view. And that would be very, very exciting. So if we think about modeling the cell with a graph or other like causal methods, we could come up with some way to estimate like some drugs that target specific parts of the graph, and that would give like the most insightful information from the causal point of view. So there are some causal discovery methods out there, which we did not talk about in that talk specifically, but we could probably take inspiration from those things. Thanks a lot for the question, Michelle.
All right. Once again, thank you everyone for attending, and thank you for host for organizing.